It wasn't until the middle part of the 19th century that the theory of spontaneous generation came under fire. And most famously, it was Louis Pasteur who conducted a series of experiments to try to disprove this theory for once and for all. Pasteur did a series of experiments that we refer to as the Swan neck flask experiments. Let's take a minute and talk about these experiments and their importance. Pasteur set up an experiment with two flasks, and inside these flasks he placed a broth solution that was full of the sort of nutrients that a microscopic organism would need, or really any simple organism would need in order to survive. So water and protein and a source of carbohydrate and so on. He took the two flasks full of nutrient broth and he sterilized the broth using heat. Now one of the flasks he left open to the air, and the other flask he plugged the opening with a wad of cotton. What Pasteur showed was that the flask that was left open grew microbes inside of the broth. Now, traditionally, anyone who believed in the theory of spontaneous generation would point to that and would say that those microbes had spontaneously generated in that nutrient broth. When Pasteur looked at the plugged flask, however, when he took some of the broth out and he looked at it under a primitive microscope, he showed that there were no microbes in the broth. And he took it even a step further. He took some of the fibers out of the cotton wad and he looked at those under the microscope, and sure enough, he saw some microbes on that cotton material. So Pasteur said, as you can see, by closing the flask, I prevented the microbes from entering the nutrient broth. They were not spontaneously generated inside that flask. They were prevented from entering. And in fact, you can see they were prevented from entering because they're here on the cotton. Now, unfortunately, Pasteur's critics said, well, in the closed flask, the spontaneous generation still happened. It just happened on the cotton instead of in the medium. If you look at the lower part of this slide, you can see some drawings that came out of Pasteur's laboratory notebooks, some drawings of the things that he saw in the open flask nutrient broth underneath his microscope. These are the little microbes that Pasteur saw in that famous experiment. Now, Pasteur was not daunted by his critics, and instead set out to repeat his experiment, but to modify it just a little bit. So what he did was he took his ordinary laboratory flask, which would have looked like this one, and he heated up that very narrow neck, that glass neck, and he bent it. So this is what we refer to as the swan-necked flask. Now the reason he did that was because he wanted to be able to leave this flask open, but to prevent microbes from getting in. So in his idea, the microbes might be able to get into this opening, but because of the bend, they would not be able to travel up and around these curves and into the nutrient broth. He actually set up two of these swan neck flasks, and then he set this one up as the flask where he did not expect microbes to enter, and he just broke this to create essentially a second flask, which he imagined would allow microbes in because it didn't have a protective pathway of twists and turns. He took these two flasks, he sterilized the nutrient broth over heat, and then he set them aside. A couple days later, He checked the nutrient broth in the open flask, and of course there were microbes growing in it. He checked the broth in the Swan neck flask, and there were no microbes growing in it. And this time, of course, there was no cotton wad, so there were no microbes anywhere to be seen. He pointed his detractors to his results, and he concluded that life cannot spontaneously generate. It can only come from life. In other words, when you find microbes inside of nutrient broth, it's not that the broth generated the microbes. It's that the microbes got into the broth. Life can only come from life. It was with Pasteur's work with the Swan neck flask experiments that we entered what we now refer to as the golden age of microbiology. The advancements that were made in the second part of the 19th century and the first part of the 20th century really advanced the science very, very quickly. Pasteur had elegantly disproved spontaneous generation. Other scientists picked up his work, and the race was on to try to figure out exactly what these things were, what these microbes that they could see under their primitive microscopes really were, and where the connection was between these microbes and the development of human disease. Now, that led to what we now refer to as the germ theory. Interestingly, Louis Pasteur gets credited with germ theory, but the history does not support that. He was actually one of many scientists who worked very hard to produce the experimental evidence that allowed for the germ theory to come into being. We give him the credit, but it was his work and the work of a lot of his contemporaries that really led to the development of this theory, which we still hold true today. What the germ theory states is that certain diseases, what we now refer to as infectious diseases, are indeed caused by tiny organisms, what we now call microbes, but what, remember, Leeuwenhoek famously called animalcules, little animals. The second part of the theory says that it's the growth of these organisms in the body of the human and other animals that causes disease. So it's not enough for them to get into the body. They then have to multiply. They have to increase in number for disease to occur. And that leads us to Robert Koch. Koch was another scientist who worked in the field of microbiology in the late 1800s. He developed a very famous system for determining what microbial organism causes which of the infectious diseases. So Koch was doing work to try to figure out for any particular infectious disease, which of these microscopic organisms was causing that disease. This systematic approach that Koch developed, we give a name. We call Koch's postulates. It was Robert Koch, in fact, who first demonstrated that a bacteria called Bacillus anthracis was the organism that was causing a terrible disease called anthrax. So let's take a look at Koch's postulates. What Koch proposed was that the first step in the identification of the microbe that causes any particular infectious disease involved isolating that organism, that pathogen, from the animal who was showing that particular disease. So you had to take body tissues or body fluids from the infected animal. you had to take that microbe into the laboratory and you had to grow it in something like Pasteur's nutrient broth. Next, you had to take that organism that you grew up in your pure culture and you had to inoculate a healthy animal with that pathogen. So an animal that was the same species, that was very important, but that was healthy, not showing any signs of disease. The inoculation might be into the mouth or up the nose or perhaps by injection, depending on the disease that was being investigated. You had to then watch that healthy animal to see if it developed the same disease. Lastly, you had to then recover that organism, re-isolate that pathogen from this newly infected animal, and go back into the laboratory and grow it in pure culture again. You then had to take that pure culture and look at it under the microscope and see if the organism that was growing looked like the organism that you isolated from that first animal. On this slide, we can see an artist's drawing of Koch's postulates. If you start up at the top, you can see step 1, which is isolating this microbe, this animalcule, this microscopic organism from an animal that was showing signs of the particular disease that you were studying. You had to isolate that organism and take it into the laboratory so it could be grown in a culture. A pure culture, in other words, you had to grow it in a way so that it was the only thing growing. It was that one organism that was growing in the nutrient broth or on an agar dish. You would then examine that pure culture underneath your microscope to see this microbe, to see this animalcule. Third, you would take that pure culture, the organism you had grown up in the lab, and you would inoculate a healthy animal with it and see if you could cause the same disease that was in that first animal. You'd then re-isolate the pathogen from this second animal. Again, grow it up in a pure culture in the laboratory, re-examine it under the microscope and make sure that it looked identical to that first organism. Now, obviously, in today's microbiology laboratory, two microbes that look the same under the microscope can be two different organisms. They can be two different species of the same type of organism. We don't only use Koch's postulates in a modern laboratory, but Koch was onto something very important in that he allowed for scientists around the world to begin connecting different types of infectious diseases with very specific types of microbes.